Hello, welcome back. In this video, we will learn a more practical way of using the visual colony characteristics that actually help the microbiologist to narrow down the possible pathogens to a limited few. Let us first begin by knowing the basics of bacterial colony. In my last video, I showed how we isolate single bacterium using various streaking methods. We cannot see single bacterium with our naked eyes, but when the same bacterium is isolated on a culture medium and incubated overnight, it multiplies to form a visible mass of genetically identical bacteria, all obtained from a single bacterium. The visible mass is called a colony and has roughly 10 raised to power 7 bacteria in it after overnight incubation. We have already learned that the blood agar and MacConkey agar are the two most common media used in our labs for plating of majority of human samples. Both of these are differential media and therefore help us differentiate bacteria based on their visual growth patterns. I suggest that before going further, you should watch my previous video to know basics of blood agar and MacConkey agar. Link is given above. So let's begin. First example is the bacteria that produce pinpoint colonies on blood agar. These colonies are typical of Streptococcus species. To differentiate between various groups of Streptococcus species, we then check the hemolysis pattern of the colonies on blood agar. Beta hemolysis is shown by group A Streptococci, example Streptococcus pyogenes, group B Streptococci, example Streptococcus agalactiae, and some of the group C and F Streptococci. Alpha hemolysis is shown by Streptococcus viridans and Streptococcus pneumoniae. Gamma hemolysis, which means no hemolysis, is shown by the group D Streptococci, example Enterococcus species. So, merely by looking at the size and hemolysis pattern, we could differentiate between various streptococcal groups before proceeding for further identification. Now, if you see large size colonies on the blood agar that is more than 2 mm size, we can think of Staphylococcus aureus, Escherichia coli, Pseudomonas species, coagulase negative staphylococcal species, Klebsiella species, Salmonella species, and even Bacillus anthracis. Here again, we can group them on their hemolysis pattern on the blood agar. Beta hemolysis is shown by three of them, that is Staphylococcus aureus, Pseudomonas species, and Escherichia coli, while others are non-hemolytic. Large beta hemolytic opaque colonies with golden yellow pigment are indicative of Staphylococcus aureus and are easy to identify. Growth of Staphylococcus is inhibited on MacConkey agar by the presence of bile salts. Large beta hemolytic opaque colonies with metallic sheen and irregular margins are seen with Pseudomonas species. Presence of fruity grape-like odor on opening the plate is another feature of Pseudomonas. Also, it produces non-lactose fermenting, that means pale yellow colonies on MacConkey agar. Large beta hemolytic smooth colonies on blood agar and large pink lactose fermenting colonies on MacConkey agar are diagnostic of Escherichia coli. Large non-hemolytic colonies with the typical ground glass appearance on blood agar and medusa head appearance on the inspection of margins using a magnifying glass is diagnostic of Bacillus anthracis. These colonies have a sticky consistency when lifted with a loop. Large non-hemolytic whitish colonies on blood agar but no growth on MacConkey agar are suggestive of coagulase negative staphylococcal species. Large non-hemolytic colonies with typical mucoid appearance on blood agar and pink lactose fermenting colonies on MacConkey agar are suggestive of Klebsiella species. We have covered pinpoint colonies and large colonies, but Proteus species shows a swarming growth on blood agar and it is difficult to find single discrete colonies after overnight incubation. This feature makes it easily identifiable. Bile salts in MacConkey agar inhibit swarming of Proteus and shows non-lactose fermenting discrete colonies on it. Proteus growth also has a typical fishy odor. Serratia marcescens produces typical red pigment on blood agar and MacConkey agar and colonies appear red on both media. A number of bacteria produce small colonies on blood agar and do not have clear differentiating features based on their colony morphology, so we cannot clearly differentiate them based on colony characters. In this video, we have used visual identification using only the blood agar and MacConkey agar. We must understand that there are variations within each species, so inspection based diagnosis should not be considered final. For further identification of bacteria, we subculture them on special media, visualize under microscopes, and do biochemical tests. These will be covered in my next videos. Do comment, like, subscribe, and share. Thank you for watching.